So it took me a long time.
How's everyone's week?
Pretty busy.
Yeah,
I'm nervous for the test though.
You're nervous about it?
Yeah, 'cause this has been like the hardest 2 sections, and now we have a test on it.
It has been pretty hard. What are you guys doing for Thanksgiving?
I'm going home.
What was that?
I'm just going back home to Bonners Ferry,
Oh, nice.
in northern Idaho.
Very fun. How about you, Jackson?
Just staying here.
Hanging out?
Yep.
What are you doing, Kevin?
Going to California tomorrow.
Is that where you're from?
Yeah, the Long Beach area.
Oh, that's cool.
Yeah.
That'll be fun.
So do you guys have any questions about the...
Well, if you want to go over like the last together.
The last one?
For what one?
Number 5.
Number 5. Okay, let's do it.
I mean, it's like I think the section was a little confusing. Like I definitely didn't get all of these problems right.
Yeah.
Yeah.
I feel like there are a couple questions where like she asks us something and then I'll go through the reading and I only find like one answer. And
I know.
like 6 answers in the answer key.
Mhm.
Like, where are you finding all of this?
I think in last week's assignment, I like wrote in the comments like, I feel like some of the stuff isn't in the reading, so I didn't know how to answer them.
Seriously.
Yeah, I did notice that.
Yeah, so I know, like Jackson last week, I didn't even do number 4 because I was like, I don't even know how to do this one. I just can't get all all the different proteins kind of confusing.
Mm-hmm. I feel like I get confused between the what was it? Like if it asks what proteins means, what sequence is, because sometimes it doesn't like specify what it is.
Yeah.
So,
I know, because I thought you had to write out like a specific sequence, but then it's like ta-ta box.
yeah.
But yeah, those are kind of confusing too. And I kind of had a hard time with like when it asked like what sequence is involved in termination. Like I was like, I just put inverted repeats because I wasn't sure if there was like a specific sequence.
Yeah.
But I did inverted repeats.
Yeah, I don't know. Yeah, so I only did 1 to 4 this week. But then I looked over 5 with the answer key.
Mm-hmm.
So I don't know if you guys want to go over that or.
Yeah, we can do that. Is there a way, like, I'm on Google Hangouts on my phone right now? Is there a way to, like, be on my computer and stuff?
You can hang up and I can re-invite you.
Okay, let's do that.
Send that.
Okay, so number 5.
Do you guys have it put up?
Sorry, what'd you say?
Do you guys have number 5 pulled up?
Yeah, I just see that both the answers.
Okay. Yeah.
I didn't really even look over 5 yet. I'm sorry, I'm so unprepared.
That's fine.
Okay. I like the old I Learn a lot better than this one.
Yeah, me too.
I'm starting to get the hang of this one, but I'm still not that much of a fan. Okay, I'm ready.
Okay. So, number A, suppose a pre-mRNA is produced by transcription, which RNA modification occurs first, and second, and last?
5N capital.
F5.
What is a modification? Like I didn't even read that part.
It's a good question.
Dude, none of us.
Do you have an answer, Jackson?
I'm trying to pull up the reading right now to see if I can find it in there.
It's at the very end, I know.
Okay.
Could I just, yeah, I didn't have to do the problem.
Yeah, it probably helps to know what the question's asking.
Okay.
First, second. Okay, mine's freezing. It's not wanting to pull up.
Oh, geez, it's like 3 pages on RNA modification.
Yeah. It says once a eukaryotic mRNA molecule is produced by the process of transcription, it can be modified in a number of ways. These changes are called RNA modifications or post-transcriptional modifications. The majority of these modifications only apply to eukaryotic transcripts. The modifications include processing, splicing, 5' capping, 3' polyadenylation, RNA editing, and base modification.
Okay. Well, I don't know. It's kind of hard because like it tells me the answer, and so I know what it is, and it's tempting just to like move on and not really.
Yeah, understand it.
Yeah,
And I'm gonna take the test and it's gonna ask a question and I'm not gonna know.
that's very true.
Let's see. So what it talks about, it says 5' capping occurs during the synthesis of the pre-mRNA transcript. It is the first modification that occurs.
Okay, so that's 5N capping.
That one basically like spelled out the answer right there.
Yeah.
It involves RNA 5' triphosphatase. Oh, okay. So 5' capping is the addition of a 7 methylguanosine. This is called 5' capping. And it's used to protect the 5' end from exonuclease attack, serve as a binding site, serve as recognition site, and regulate splicing.
Okay.
Correct.
Regulate space. So we know that that one is the first one to occur.
Yeah.
So the second RNA modification is a 3' end polyadenylation.
So it is not, oh, so you go ahead.
Okay. This says that multiple adenine residues are attached to a site near this end. So I don't know how that helps.
Let's see. It functions to increase the stability of the mRNA. It terminate transcription in eukaryotes, helps to facilitate the transport of mRNA from the nucleus to the cytoplasm, and enhance the recognition of the mRNA transcript by the ribosome.
During translation. Okay.
And polyadenylation occurs after 5' capping, but prior to RNA splicing.
Okay.
So you can assume there, I guess, that it's second.
Mm-hmm.
And then because of that, intron splicing is last. Okay.
Yep. Except here it says RNA splicing, but the answer key says intron splicing.
Yeah, that would confuse me.
I'm not seeing anything. Oh. I'm trying to see where it talks about introns.
It says that so initial transcription produces a pre-mRNA molecule, and this is then spliced, and that's called an intron. So I think it's basically the same thing.
Perfect. Perfect. So then the next, do you have any questions on part A then?
No.
Nope.
All right, so part B, ribozymes play an important role in RNA modification. What is a ribozyme, and which RNA modification process involves ribozymes? Look up ribozyme. So it's a catalytic pre-mRNA.
Yes.
And it catalyzes the removal of its own intron.
Okay. So intron splicing is the process that involves ribosomes.
Right.
Okay. So do you guys have questions about number B? The letter B, I guess.
Nope.
Okay, so C, what is the function of each of the following molecules in RNA modification? SNRNP.
Small nuclear ribonucleoproteins.
They are composed of uracil-rich small nuclear RNAs that are about 100 to 200 nucleotides long and proteins.
Yeah.
They catalyze the enzymatic cleavages responsible for removing introns.
Or introns, so introns are a bad thing?
Are they?
No, either. Talks about removing the intron, so
Yeah,
Like you're always trying to remove them.
it says that it's removed and discarded. So introns you want to get rid of, but then exons are used to assemble a mature mRNA.
Yeah. It says when the DNA contains an intron, between the template DNA and the mature mRNA, it forces the intron DNA sequence to loop. I guess you don't want it to loop.
Guess not.
Okay. Okay, moving on. Guanosine nucleoside.
So for guanosine nucleoside, it says it serves as a cofactor for intron splicing.
the group 1 intron splicing.
Yeah, group 1.
Okay.
That's it for that.
CPSF.
CSF. And for the cleavage and polyadenylation specificity factor.
Specificity.
That is a mouthful.
Specificity.
Specificity.
It's a hard word to say.
CPSF.
And so these cleave to the RNA 10 and 35 nucleotides downstream of the recognition site.
Cool.
So that's its main function. So
Yeah, that's a lot more in depth than what it's telling us here.
Mm-hmm.
I wonder where she's getting all those other words or descriptions from.
yeah, I've always wondered that. So poly-A polymerase.
Attaches multiple adenine residues to the 3' end of the transcript.
Yes.
As many as 250.
So it adds 250 A bases. Okay. And then the last one.
Guanylyl transferase.
Guanylyl transferase.
Right.
So poly-A polymerase is in the 3' end, and then guanylyl transferase is the 5' end.
Okay. Recognizes and hydrolyzes GTP to produce GMP and pyrophosphate.
All right. Do you guys have any questions on those?
No?
This is hard.
I mean, it's pretty straightforward when you're reading it, but trying to remember all that.
That's gonna be tough.
I know.
I liked genetics because it was kind of like math, and then this is like memorizing facts.
Yeah, that was a lot easier, I feel like.
What are you guys going into? We already had this conversation.
I don't think so.
I don't think so.
I'm doing human biology, going into physical therapy.
Okay.
Nice. I'm going into dentistry.
See, I was originally dentistry, and then I recently changed last semester to physical therapy.
Oh, really?
Yeah.
Physical therapy. You have to take kinesiology?
What's that?
You have to take kinesiology?
Yeah, that's what I'm trying to get into right now for next semester. I'm number 49 on the wait list.
Is it,
Got a chance.
yeah. Is that with Brother Perks?
Yeah,
Yeah, I'm taking that class right now. It's hard.
I bet.
Yeah. What are you going to, Lizzie?
Cosmetic science.
Nice.
So like making makeup and stuff.
Making makeup.
Yeah, I definitely don't want to be a doctor or anything because don't have the drive to do that. But either cosmetic science or I love like anatomy. So I think it'd be cool like an anatomist or something too. But
I didn't know they even had cosmetic science, but I guess somebody has to.
yeah. Yeah, well I love makeup and love science, so I thought I'd combine the two. And there's a career in there, so
The company I work for has a bunch of those people, cosmetic scientists that work for us, and then I test all the products for them.
no way.
Yeah, I'm working in a microbiology lab, and so our R&D group designs and formulates all the new makeup that we sell and
hmm
supplements and things like that, and then I test it for bacteria and it's safe.
Wow, you're like a real scientist.
Damn,
Pretty tech.
that's impressive.
Yeah, I did that as my internship last semester, and they hired me on afterwards.
Dang.
Awesome.
So that worked out really well.
Yeah, that's cool.
Kev, are you growing a mustache?
Yeah.
Are you doing No Shave November?
No, it's kind of hard to see.
I can see it. It looks good. That's like as much as you can grow at BYU, huh?
That's right. That's all you can grow.
Mm-hmm. Dang. Well, at the end.
Yeah, that was a good meeting. Because yeah, the very last question says that she'll prepare a drawing.
She
But
says that, but I never see drawings.
yeah, I feel like she says that a lot, and then there's never a drawing.
Mm-hmm.
So
Oh, I just feel like I'm gonna do so bad on this test.
I didn't know there was a test until you said that, so I'm excited for that.
I'm trying to go to Seattle tomorrow, and so I have to take it tonight.
it closes tomorrow, right?
Mm-hmm. And I don't have to worry about it tomorrow, so I'm just gonna try and get tonight.
Yeah. Well, good luck on the test, guys.
Yeah, you too. Have a good weekend. Have a good Thanksgiving. Do we have to meet next week? Probably.
I don't know.
Even though it's Thanksgiving.
Here, let me check.
I'll send her a message on this board. She usually responds to my posts pretty quickly when I post her questions and stuff.
Okay.
I guess we'll just let each other know.
Yeah.
Well, have a good Thanksgiving. Good luck on the test, guys.
You too.
You too.
See ya.
Hey, bye.